Doctors, today we will learn about the Krebs cycle. Each step very simply we will understand. Let's see. First is introduction. Citric acid cycle or TCA cycle is also called as Krebs cycle. Most important for metabolic pathway for energy supply to the body. Essentially involves oxidation of acetyl CoA to CO2 and H2O. It is the central metabolic pathway and final common oxidative pathway for carbs, fats, and amino acids. It connects almost all individual metabolic pathways. It provides intermediates required for synthesis of amino acid, glucose, and hemoglobin. Next, the definition of the Krebs cycle. It is the central metabolic pathway in aerobic organisms that oxidizes acetyl CoA into CO2 and H2O. This process produces ATP or GTP and reduced coenzymes that NADH and FADH₂ for use in ETC. It is very crucial process for generating ATP for the body. Next we will discuss about the site. Tissue site, it is the almost all the tissues of the body, high activity in muscles and liver. And intracellular site, mitochondrial matrix as an enzyme are present there because all the enzymes are there in the mitochondrial matrix. Then the starting products, acetyl CoA and oxaloacetate. Then end products 2 mole of CO2, 1 ATP or GTP, 3 NADH and 1 FADH₂, and finally oxaloacetate is regenerated. How? Next we will see. Pathway. The main consists of 8 sequential enzymatic steps. Let's see steps one by one. Step 1, formation of citrate. Krebs cycle proper starts with the condensation of the acetyl CoA and oxaloacetate. Enzyme is required citrate synthesis and formation citrate. It is the irreversible step. We will see next step is second and third step. Citrate is isomerized to isocitrate and citrate first citrate forms cis-aconitase by the dehydration, and cis-aconitate is finally form isocitrate with the help of dehydration. In the both two steps, aconitase enzyme is used in both the reactions. Next we will see fourth and fifth step, formation of alpha ketoglutarate. Isocitrate convert to oxalosuccinate and oxalosuccinate converted into alpha ketoglutarate. Both reactions required single enzyme, that is isocitrate dehydrogenase. And first one, isocitrate converts oxalosuccinate with the formation of NADH and liberation of CO2. It's also called as the oxidative decarboxylation. Next we will see the sixth step, conversion of alpha ketoglutarate to succinyl CoA. Alpha ketoglutarate is converted to succinyl CoA. It's also called as the oxidative decarboxylation. Enzyme is alpha ketoglutarate dehydrogenase complex. It is the irreversible step. And this enzyme complex contains 5 cofactors, that TPP, lipomide, NAD+, FAD and CoA. Mechanism of reaction is analog to conversion of pyruvate to acetyl CoA. While conversion of pyruvate to acetyl CoA, there is the PDH complex is there, and here it is the alpha ketoglutarate dehydrogenase complex. It is just analog to the PDH complex. Next we will see the seventh step, formation of succinate. Succinyl CoA is converted to succinate using enzyme succinyl thiokinase. phosphorylation of GDP to GTP or ADP to ATP. This substrate level phosphorylation. This is, remember, this is the substrate level phosphorylation. Two isoenzymes of the succinate thiokinase. First one is dependent on GDP and second one is dependent on the ADP. Dependent on GDP, that is the gluconeogenic tissues present there in liver and kidneys, and dependent on ADP, it is the non-gluconeogenic tissues, that is brain and heart. Next is 8th step, that the conversion of succinate to fumarate. Succinate converted into fumarate with the help of succinate dehydrogenase. That's the production of FADH₂. And step 9 is formation of malate. Fumarate converted to malate using fumarase catalysis. And step 10 is conversion of malate to oxaloacetate. Malate converted into oxaloacetate with the help of malate dehydrogenase enzyme. Then the synthesis of NADPH. Oxaloacetate is regenerated, combines with another molecule of acetyl-CoA and continues the cycle. In this way, oxaloacetate is regenerated. Next, we will see the summary of the Krebs cycle. Acetyl-CoA plus 3NAD plus FAD plus GDP plus inorganic phosphate plus 2 water molecules finally forms the 2CO2, 3NADPH, FADH2 plus GTP and COA. In this way, acetyl-CoA is converted into carbon dioxides and the source of energy. Let's see the energetics: 3 NADH, 1 FADH and 1 ATP or GTP. Finally synthesized, these addition total 10 ATP from 1 acetyl-CoA. Regulations mainly done by the enzymes and the levels of ADP. Let's see how by enzymes. First one is citrate synthase inhibited by ATP, NADH and long chain fatty acyl-CoA. And second enzyme isocitrate dehydrogenase activated by ADP, inhibited by ATP and NADH. And third one is alpha ketoglutarate dehydrogenase inhibited by succinyl CoA and NADH. Next one is level of ADP for the next fourth one is the availability of the ADP. Level of ADP, if there is low or high, let's see what happens. The importance for the cycle to proceed, and unless sufficient level of ADP are available or oxidation of NADH and FADH2 through ETC stops. It is the availability of ADP, it's important. Their accumulation leads to inhibition. So unless sufficient level of ADP are available, oxidation of NADH and FADH2 through ETC stops. Other factors, then fifth point is our hyperammonemia, characterized by elevated ammonia found in circulation and then found in severe liver diseases and genetic defects of the urea synthesis. Accumulation of ammonia withdraws alpha ketoglutarate to form glutamate and glutamine. Reduction in levels of the Krebs cycle intermediates that therefore that decrease in ATP generation. Ammonia inhibits that alpha ketoglutarate and pyruvate dehydrogenase enzymes, and there is therefore the loss of the consciousness, coma, convulsions may be even fatal. Then we will see the significance of the Krebs cycle. First one is amphibolic nature of the TCA cycle. It is the both catabolic and anabolic, hence it's called as the amphibolic pathway. Next is actively involved in gluconeogenesis, transamination and deamination. Important anabolic reaction connected to TCA are: first one is succinyl CoA used for synthesis of porphyrin and heme, then Mitochondrial citrate transported to cytosol where it is cleaved and gives acetyl CoA for biosynthesis of fatty acid and sterol. This we will see in the fatty acid synthesis. Synthesis of aspartate and glutamate that have precursors oxaloacetate and ketoglutarate, alpha ketoglutarate. Aspartate and glutamate required for synthesis of non-essential amino acids, purines and pyrimidines. In this way, the pathway is amphipholic. Then it is the anaplerosis, anaplerotic reactions. Definition that reaction consists to replenish or to fill up intermediate of the TCA cycle are called anaplerotic reactions. Salient features of the important anaplerotic reactions, that first one is the pyruvate plus CO2 plus ATP. It gives oxaloacetate plus ADP with an organic phosphate with the enzyme pyruvate carboxylase. It is the ATP dependent carboxylation reaction. And next, the pyruvate plus CO2 plus NADPH plus H plus, it gives malate plus NADP plus H2O with the help of malate dehydrogenase. Third one is the transamination reaction, process in which amino acid transfers its amino group to keto acid and itself get converted to keto acid. Formation of alpha ketoglutarate and oxaloacetate occurs by this mechanism. Next one is the glutamate plus NADP plus H2O that gives alpha ketoglutarate plus NADPH with the help of glutamate dehydrogenase enzyme. Let's discuss the inhibitors of the TCA cycle. Enzymes and their inhibitors are there. First one is iconitase enzyme. That inhibitor is fluoroacetate in the form of non-competitive inhibitor, and alpha ketoglutarate dehydrogenase arsenate is the inhibitor in non-competitive form. Succinate dehydrogenase have malonate as a competitive inhibitor. Let's see some little bit information that for fluoroacetate, it is the suicide substrate regarded as a poison in some plants, used as anti-cancer drugs and some pesticides. This is the Krebs cycle. We will overlook acetyl CoA combines with oxaloacetate with the enzyme citrate synthase formation of citrate. Then these two enzymes forms isocitrate. Then isocitrate forms oxalosuccinate with the enzyme of isocitrate dehydrogenase, and oxalosuccinate form alpha ketoglutarate with the enzyme of oxalosuccinate decarboxylase. And here it is the substrate level phosphorylation, that succinyl CoA converted to succinate with the enzyme of succinyl CoA synthetase. It is the formation of GTP. Here formation of FADH₂ when succinate is converted into fumarate with the enzyme succinate dehydrogenase. And finally malate converted into oxaloacetate, and oxaloacetate is regenerated. If you like it, my handwritten notes for Krebs cycle, and if you like it, share, like and subscribe. And thank you. We will meet in next video.